mitt navn Hans Petter Graver. Jeg preses i Videnskaps-Akademiet, og det en glede for meg å ønske velkommen til dette webinaret om regenerativ medisin. Dette webinaret ledd i Akademiets arbeid med vitenrådgivning, eller Science Advice. Dette arbeidet med vitenrådgivning en viktig del av Akademiets aktiviteter. Det utgjør en av 3 hovedpilarer, og vi mener at dette viktig både fordi Akademiet representerer en stemme som uten institusjonelle bindinger og avhengighet, så vi slik sett mer uavhengig på en måte enn universiteter og forskningsinstitutter, fordi vi ikke lever av midler til forskning. Og for det andre fordi vi jo gjennom våre medlemmer og gjennom vårt kontaktnett også har et stort nettverk av fremragende forskere. En viktig del av Science Advice-arbeidet samarbeidet på det europeiske planet med også europeiske akademier. Og et av dem jo EASAC, som et europeisk Science Advice-nettverk av akademier, som arbeider systematisk med utarbeidelse av rapporter som skal tjene som grunnlag, som innspill til offentlige beslutninger i de forskjellige landene. Og i dette tilfellet så har vi jo en rapport fra EASAC som ligger til grunn for det tema som oppe i dag, men jeg skal ikke ta på meg å presentere det, for det det andre som skal gjøre, så jeg vil bare si velkommen til webinaret, og jeg regner med at vi alle får en interessant og utbytterik seanse gjennom foredraget og diskusjonen, så takk og velkommen.
Ja, da sier jeg takk til deg, Hans Petter, og velkommen til alle som med på for å høre på dette. I'm going to switch to English for a little bit. We're probably going to have we'll probably be going back and forth between English and Norwegian a bit through the webinar. We have international participants. So I'll introduce the theme in English. I think it's important to say that regenerative medicine is a field that is developing very rapidly, and this is fueled by advances in quite a few different areas of research: human stem cell biology, biomaterials, biofabrication, sensor technology, and others. And these are all converging on a common goal, which is to reconstruct or construct de novo tissues and organs that have been damaged or destroyed by injury or disease. It's a very multifaceted field, as you will learn during this session. And as with all new technological advances in medicine, there is a promise that there will be real advances in medical treatment. And there are also poorly founded expectations that the technology will advance more or faster than it can. And the balance between the hope and the hype is important. And in fact, in this case, it's urgent because there are opportunists who would try to take advantage of patients who are desperate for a cure and believe that regenerative medicine may be the way to get there. So this led the European Academy's Science Advisory Council and the Federation of European Academies of Medicine to, see if I can get my PowerPoint to advance here, to commission a panel of experts to write this report on the status of regenerative medicine, including its regulation. And it's this report that has motivated today's webinar. We have a panel of international Norwegian experts in the field with us today. And since web cameras are not always kind, I decided to present pictures of them here. Olle Lindvall, these are about in the same sequence they will appear during the webinar. Olle Lindvall from Lund, Tony Atala from Wake Forest, Rune Kjeken is from the Norwegian Medicines Agency, Statens Legemiddelkontroll. Kamal Mustafa from the University of Bergen. Goran Petrovski from Oslo University Hospital. Helge Ræder from University of Bergen. And Hanne Scholtz from Oslo University Hospital. Myself and Morten Moe, also from Oslo University Hospital. And then we have Ole Johan Borge at the top here. He's going to be leading the proceedings. He has a master's in biotechnology from the University of Oslo, a PhD in stem cell biology from Lund University. He's worked for many years in the Norwegian Biotechnology Advisory Board, first as a senior consultant and later as director. He's also had a period as special consultant and department head at Innovation Norway. And since this year, he's head of the Department of Health, Research and Innovation at the Norwegian Research Council. I think it's fair to say that he knows this field pretty well, and he's certainly well qualified to lead this webinar. And on that note, I will pass the baton to him. Ole Johan, welcome. Thank you for taking this, this task, and off we go.
Thank you very much, Joel. And I have been very much looking forward to this day. So I think without any delay, I would like to present the first presenter immediately. And I'm happy to introduce Olle Lindvall. Lindvall received his PhD in 1974, earned his medical degree in 78, and became professor in clinical neurology at the Lund University Hospital in 92. Lindvall was leading the clinical transplantation program for Parkinson patients, the famous one, as I guess most of you know, in Lund between 83 and 2012. This program pioneered cell replacement strategies and was the first to demonstrate that transplanted neurons can survive, grow, and become functional or integrated, and also give rise to improvements in the diseased adult human brain. Lindvall was the clinical coordinator of the EU-sponsored project called EuroStemCell from 2003 to 2007. And he has been a board member of the International Society of Stem Cell Research, the ISSCR, from 2004 to 2011. As we just heard, Olle here is invited to present the report, Challenges and Potential in Regenerative Medicine. And as you can guess, Lindvall was or has been involved in this work as an expert to prepare this report. So, Olle, the word is yours.
I'm very grateful to be able to participate in this and to have been invited. What I will try to do is to give an overview of the main messages in this report. Of course, this is a 30-page report, so I cannot describe all the details. So the question is, why is there a need for a report like this? Well, Joel mentioned that we all know that stem cell and gene-based therapies intended to regenerate damaged and diseased human tissues hold great medical promises. And for the first time, there is a possibility that we could develop novel therapies for human conditions which currently lack treatments. But there is an increasing problem of commercial clinics offering unregulated and unproven therapies. Because if we look at the field, there are very few diseases where there are stem cell or gene-based therapies which with proven efficacy. And there is another alarming trend which one can recognize, and that is that to lower the scientific requirements and evidence for clinical application from regulators. It is a problem that the high hopes from desperate patients and relatives, and also from the biotech industry, can put pressure on regulators to accelerate authorization of stem cell-based approaches. And this, of course, put patients at risk. So why is this responsible translation of stem cells to the clinic so important for success in patients? Well, of course, first, to avoid the risk for major adverse effects. If you move too quickly without a solid experimental basis, there is a risk for major adverse effects. And I think the best example of this is perhaps what happened at Karolinska Hospital when the surgeon Paolo Macchiarini took a synthetic trachea. They were, and together with some stem cells, they were implanted in patients. And that was carried out without solid evidence from preclinical animal models that this could work. And there is another important thing here, and that is you have to optimize the chances for beneficial effects, and you have to avoid risk for lack of improvement. If you take a stem cell-based approach and move it too quickly without proper design, the risk is that a potentially competitive, potentially very promising therapy would be regarded as not working because it was not done in the right way. And both these things, major adverse effects, that it doesn't work, it seems that it doesn't work, will lead to major delays in the development of clinically useful therapies. So what Joel mentioned is that this report was an initiative from the European Academy Science Advisory Council and the Federation of European Academies of Medicine. And under the chairmanship of a professor from Germany, we were experts from 11 different European countries nominated by member academies. I was nominated by the Royal Swedish Academy of Sciences, and this report was written then for a target group, European Commission, European Medicines Agencies, the EMA, the European Parliament, member states, academies, and international policy makers and stakeholders. And the group identified 3 major problems with the application, with patient application of stem cells. I will focus on stem cells since that's my own field. 3 major problems. One is that stem cell-based approaches have been moved to clinical application in patients, often with a weak experimental basis, not sufficiently well tested for efficacy in preclinical models. And that has been done without understanding the mechanism of improvement of the stem cells and the pathophysiology of the disease. And the problem here is that I think it's quite well established that stem cells can act based on different mechanisms. For example, when you put stem cell and stem cell derivatives into the brain, they cannot just replace injured neurons or glial cells, but they can also modulate inflammation, they can have trophic effects. And if you are going to move stem cell to the clinic, you have to have better understanding of why there is an improvement and how to optimize that. And also, what is the disease? What is in the disease that you would target with your treatment? And also, I think that we identified that there is a limited understanding that without a solid experimental basis, even well-planned and well-performed clinical trials are meaningless. I mean, if the experimental basis is poor, even if you do a careful clinical study with proper controls and so, then it becomes meaningless. So, and then there is the problem, which was also mentioned initially, about unproven therapies, what we call stem cell tourism, that the patients travel to clinics, so-called clinics, which offer stem cell treatments or treatments with stem cells, which are unproven, and they do that with high cost. And this is just one example, which is called a company called Swiss Medica, which offer stem cell treatments for, in principle, all the diseases you can think of, including Parkinson's disease and stroke. And I can tell you that there is no stem cell treatment for Parkinson's disease and stroke, but they are operating in Austria, in Switzerland, in Serbia, in Slovenia, and in Russia. But in this context, I think it's very important to emphasize that when we are talking about travel for stem cell therapy, that patients and relatives are never to blame. Who can blame a person, a desperate person or patient who wants to have a treatment if there is any chance of success? But clinics which offer these unproven therapies, and when patients have to pay, that is of course completely unacceptable. When safety, efficacy have been proven, then of course stem cell travel is acceptable. So what are then the main recommendations and key messages from the group? Well, it is a big problem, as we said, that stem cell in the stem cell field, that stem cell based approaches are moved to the clinic with weak experimental basis and with poor understanding of the pathophysiology of the disease. So a solid experimental basis is absolutely necessary for clinical application. And in the report, we strongly strongly recommend better preclinical research, including improved understanding of the pathophysiology of the disease. It should be emphasized that stem cells, unlike drugs, stem cells do not have a precisely definable molecular and chemical composition, which means that stem cell products prepared in different laboratories may vary, and they can also vary at different times in the same laboratory. And factors contributing to stem cell variability and issues for product standardization have to be identified. When a stem cell product is going to be used in patients, it is of course absolutely necessary to test its efficacy before delivery to patients. It's also important that identify safety criteria for administration of stem cells to rule out the risk, or at least minimize the risk for tumorigenicity and other adverse effects. And finally, one can discuss how much, but I think studies to understand mechanism of action underlying efficacy of stem cells, that is very important. You have to understand what the stem cells are doing. But not only preclinical research has to be improved, but also clinical research, including understanding of valid outcome measures. It is important that, unlike drugs, which can act almost momentarily or within minutes or hours and last for one day or a few days, cells, it may take some time before they start to work, and they may work for a very long time. In fact, if you put cells into the brain, they may you may not see anything until 3 months after implantation, and maximum effect could come after 1 to 2 years. So and then, of course, factors contributing to clinical response variability, including which patients could actually improve. I think that will be crucial. This will be more personalized medicine with these approaches. Stem cells will not work for all patients with Parkinson's disease, for example. It's not a magic cure for everything. And finally, it is important that when performing clinical trials in regenerative medicine, the design and monitoring standards should adhere to similar principles as for other clinical trials, for example, with drugs. It is important, and that was stressed by the committee or the working group, that how important it is that preclinical and clinical researchers follow recommendations from scientific societies such as the International Society for Stem Cell Research. I was involved and was co-chairing the committee when the first recommendations were produced now 12 years ago. And I think those are very, very valuable also for your discussion later today. The group underscores the importance that a close interaction between science and regulatory authorities. Cells and genes, they're completely new approaches for treatment. And I think it's very important. We all want these approaches to reach the clinic as quickly as possible, not too early, not too late for our patients. But so there has to be an interaction between science and regulatory authorities to promote quality, safety, and efficacy, and to generate sufficient robust evidence while taking care not to jeopardize capacity in early phase research. So research should be stimulated, interaction, and exactly when at the optimal time should be moved to the clinic, should be subject to authorization. And I think this is very important because there are, as I said, a trend of accelerated approval, both from the point of view of companies and patient organization. There are companies which want to move poorly efficacious approaches to the clinic. And that can also induce patient organization to lobby for marketing authorization. And in fact, as you may know, accelerated access and conditional approval mechanisms have been introduced in the US and Japan. And in the United States, the potential therapeutic value of stem cells has been used politically in calls for their regulation. And the proponents for their regulation, they suggest that the regenerative medicines should be allowed onto the market based on just proof of concept and safety testing. But as we all know, a phase 1 clinical trial with safety testing, what that tells us is just that it's possible to continue testing in, it doesn't allow a major testing in many patients, but just to continue to work with this approach. And there is also a good example from Japan when this accelerated market authorization was implicated with a application of stem cells for spinal cord injury, and with quite poor understanding of the nature of the cells, and also with the weak, the clinical data were also quite weak. So this is a problem. And finally, it is important as the group emphasized, to urge EU member state regulatory authorities to exert tighter control on unscrupulous providers up to the closure of clearly fraudulent website to stop or suppress as much as possible stem cell tourism and this clinic. It's not easy, but it is absolutely necessary. It's also important that healthcare providers address challenges with stem cell treatments at high cost. They may require one treatment or several treatments. It may be necessary to follow patients over many years to really evaluate the cost benefit. And that has to be done at an early stage when the evidence base is still being developed. And it is, of course, something to discuss. High cost effect accessibility, and raise the issue of accountability. Who decides about access to these quite expensive treatments? It is also a problem that there is in this phase for going from basic stem cell research up to clinical application. It is a very expensive phase, to clinical translation, to run the clinical trials. And it has to be a long-term commitment from research sponsors. And therefore, it is important to explore how to attract long-term financial support from EU and others for stem cell research and its clinical translation. I don't think this can be done without any involvement of the industry. And I think it is important to find ways of interaction between the industry, between companies and academia. It's also important that the health services institutions are involved in the clinical research very early and its translation to clinical practice. Health services institutions have can enable clinical trials, access to patients, and I think one important aspect is to develop health economic models for cost-benefit assessment of this treatment. And to do this at an early stage is also very important to promote the development of this. And finally, the committee emphasized how important it is to improve professional education and training, to introduce regenerative medicine in the curriculum. I don't think it is there at the moment. And to teach on professional guidelines and ethical, legal, and social issues related to regenerative medicine. And finally, improved public and patient communication is crucial. Independent and evidence-based sources of information should be promoted so that patients and their families can compare different treatment options. There is a very good, ISSCR has a very good homepage where you can actually, a website where you can actually have questions and answers, you can get a lot of information. And my own experience, having worked with cell therapy since almost 35, 40 years, no, I get many, many questions, letters, emails from patients and relatives. And I think appropriate, honest information sometimes is the most important thing you can do for them. And they would then not go for stem cell tourism. I think that they know that they have done everything they can. But I wanted to just now give you one example from our own work, namely to try to develop a treatment based on transplantation of stem cell-derived dopaminergic neurons into the brains of patients with Parkinson's disease. And I take this example to illustrate that even if the potential of regenerative medicine and stem cell-based approaches is great, in my view, the road to clinically effective stem cell treatments and other treatments in regenerative medicine will probably most likely be long and winding. As you know, the main pathology in Parkinson's disease underlying the motor symptoms is a loss of dopaminergic neurons in the midbrain, leading to a loss of dopamine in putamen and caudate in the striatum. And this loss of dopamine leads This leads to hypokinesia, namely poverty and slowness of movement. Sometimes there is no movement at all. Shaking hands, legs, rigidity, muscle stiffness, but can also lead to other symptoms like which are not motor symptoms, like depression, dementia, autonomic disturbances. And this is explained by that the pathology does not only affect nigrostriated dopamine neurons, which underlies most of the motor symptoms, but also non-dopaminergic systems in other areas of the brain. And our main treatment since almost 50 years is L-Dopa, which is converted to dopamine. And then there are also other medical treatment. And now in a more advanced stages of the disease, deep brain stimulation. But in conclusion, there is a need for novel therapeutic approaches in Parkinson's disease. And the 41 years ago, actually now, two studies were published which triggered this field. And these studies showed that a brain tissue containing dopaminergic neurons from rat fetuses, which was implanted into the brain of adult rats, ameliorated the symptoms of experimental Parkinson's disease. So the question was, of course, could you take dopamine neurons from the fetal brain, put them into the brain of Parkinson's patient, and replace the dopamine neurons which had died? Could these findings be translated into cell therapy for Parkinson's patients? And over the coming years, many laboratories were working with this, trying to improve the methodology and move towards the clinic. And one important finding was when we could show that dopamine neurons taken from aborted dead human fetuses aged 6 to 9 weeks post-conception, if they were implanted into a dopamine-denervated rat, a model of Parkinson's disease, they survived and they formed connections and into the denervated stratum in the denervated target area, and they also improved the motor behavior of the animal. So this was this led to the application in 1987 of this methodology, and just a suspension, clumps of dopaminergic, dopamine-rich tissue from the mesencephalon of aborted human fetuses, dead fetuses, were then injected stereotaxically into the brain, into the putamen and caudate nucleus. And after 2 years, in the second round of transplantation, we could actually show that it was possible to get survival of these cells in a patient with Parkinson's disease. These just are positron emission tomographic images horizontally through the brain. And you can see that there is an area here which was not seen before transplantation, which shows that the transplant has survived. That was later been confirmed with histopathology. And this is of our 18 patients. There were other patients operated in other laboratories and clinics. This is one of our best patients. We had 4 patients which could stop L-Dopa treatment completely. And this is one patient who 10 years after transplantation is without L-Dopa treatment since 3 years after transplantation. He has a normal uptake of this tracer, fluorodopa. So on the grafted side, he has a normal dopamine innervation, which releases dopamine normally. But on the side which was not transplanted, there is no uptake of this fluorodopa. And interestingly, when he died 24 years later, when he was over 80, we could show that on the non-grafted or grafted side, there was no dopamine innervation at all. On the grafted side, there was a rich, near normal dopamine innervation extending throughout the striatum. and there were tens of thousands of dopaminergic neurons. So this is a quarter of a century after implantation of these cells. But this is, of course, important because it shows proof of principle that cell replacement can work. Grafts can re-innervate the innervated areas, become functionally integrated, release transmitter, dopamine, and restore function in the diseased 50, 60 years old patient's brain. But there are problems with human fetal tissue. It's only available in limited amounts. Only few patients can be operated. There was a variability of functional outcome due to poor standardization. Some patients did not improve at all. Some patients showed a quite dramatic improvement lasting for years. And then, as some of you may know, there was some involuntary movement in some patients, which was troublesome. So what the field needed and need is a possibility to generate large numbers of dopaminergic neurons, standardized quality control. And here stem cells come in. And now we know that dopamine neurons for patient application can be generated both from human embryonic stem cells and iPS cells, induced pluripotent stem cells. Here you have the what we did before, namely the fetal mesencephalon. We took from the dopamine-rich tissue here and implanted in patients the dopaminergic progenitors. You can take from the blastocyst the embryonic stem cells and generate dopaminergic neurons in different ways. But you can also, from somatic cells like skin cells, produce induced pluripotent stem cells and then differentiate them to dopaminergic progenitors. And there are three main key findings, key scientific findings on the way now to patient application. And the first one was that Lawrence Studer's lab in New York, they showed now in 9 years ago that if dopaminergic neurons could be efficiently generated from human embryonic stem cells, and the dopaminergic neurons could re-innervate the stratum of the rat and have functional effect without tumor formation after transplantation. And then Malin Parma's group, they showed in 2014 that the human EASAC-derived dopaminergic neurons, they survive and have similar comparable functional effects as fetal ones after transplantation into rats, which was promised that they may have the same effect as we have seen with fetal cells with these stem cell-derived cells, which is, of course, very important for how we design clinical trials now. And then Jun Takahashi's group showed 3 years ago that you can take human EPS cell-derived dopaminergic neurons, and they can also survive and function without forming tumors after transplantation in monkeys. At the moment, these studies from Studer's lab, studies from Malin Parma, including our work in Lund, we are planning these clinical trials will start next year or the year after, whereas already 4 patients have been operated in Japan with this procedure. So what is will be the major scientific questions in the clinical trials with stem cell-derived dopaminergic neurons in Parkinson's disease? Well, the first is, of course, whether this could be done without major short and long-term risk. I think everything has been done to avoid tumor formation, for example, but to avoid major short and long-term risk. So safety will be the first objective, of course. Then, of course, it is, and since this is, the whole idea is that the transplant should be able to replace the lost dopaminergic neurons. and to replace them and to provide a new dopaminergic innovation in the brain. That's the whole idea. And then we have to assess what is happening with that. Can stem cell-derived dopaminergic neurons really do that in the human brain? That is unknown at the moment. And then, of course, even if it's a safety trial, one would look for evidence of efficacy. But that to show efficacy, we need, of course, to have optimize the procedure and of patient selection in subsequent phase 2 and 3 clinical trials, which would most likely involve a sham surgery control. But if this works, what can we then hope to see? Well, the improvement of motor symptoms, but this will not be a cure for Parkinson's disease. And that's very important to emphasize that. The patient's intrinsic neuron systems, for example, serotonergic, cholinergic, will continue, and these dopamine-resistant motor features and non-motor symptoms will most likely not be improved. So I think it's very important to understand that this is targeting a specific area of the brain, and other areas of the brain, pathophysiology related to those areas will not be influenced. And it will be clinically competitive, even if the motor improvements outweigh the worsening of non-motor symptoms for patients. So patients have to feel that this was worth it, even if they have some continue to have non-motor symptoms. So what can we conclude then? What I wanted to show you is how important this is with patients here, and that long-term scientific and clinical efforts will be needed to move these approaches to the clinic. And there is no fast track for stem cells to clinical application. Hard work, long-term commitment, excellent science, close interaction between researchers and clinicians, and high ethical and regulatory standards are the only way to success. And let me end by saying, yes, that there will be ups and downs. Sometimes it works beautifully, sometimes the field will struggle. But as Winston Churchill once said, success is not final, failure is not fatal. It is the courage to continue that counts. Thank you very much.
Thank you very much, Olle. And I trust that the applause would have been easily heard if everybody had their cameras and microphones on. I would now very much like to invite both the panelists, but also the audience, to either raise your arm or write your name in the chat so you can ask questions. We have now 15 minutes for questions and hopefully answers from Olle. I see one question from Goran Petrovski. Goran, please.
Thank you for the nice presentation. Is safety alone or and efficacy, which are important to make a clinical trial worthy for regulatory authorities? For many times, they're talking only about safety in some of the EU grants.
Oh, I agree that that's from the regulatory authorities. I think that Joel will have had some direct contacts with one of our colleagues working with this aspect when it comes. But that's also my experience, that safety is very much what they look at. And I think that, on the other hand, for us working in the field, of course, efficacy is crucial. It may be that, for example, in a disease like Parkinson's disease, where there is quite a lot of treatments already available, like L-DOPA, but also deep brain stimulation and infusion into the intestine of DOPA. I think that the requirements to move to the clinic when it comes to efficacy, to show efficacy, is of course higher than if it is an intractable disease. But it's also my experience that regulatory authorities look very much on the risks and adverse effects.
Thanks.
Then I would like to have Hanne Scholtz ask one question, and thereafter Morten Mo. So Hanne.
Yes, thank you very much, Olle. It was a very nice presentation. I just wonder, you show evidence of these unproven stem cell clinics that pop up around us, and how should we act to them? Do we have any tools, or how should we correct them so we can have a safe and good development of the stem cell?
If there would be a good way of doing this, but unfortunately there is not. I think that this is something, when you're developing your own strategy here, I think you have to interview those who have been working directly with this. I think ISSCR, where I was active a lot, but now not lately, on this topic, tried to, and they put even this websites, they put that on their own website as examples of stem cell clinics offering these unproven treatments. The problem was that there is so much money in this, so that these clinics, their lawyers threaten the ISSCR with, in principle, getting ISSCR bankrupt due to lawsuits. So I think that this is a very, very delicate way of doing it. And I think it has to be done in interaction, probably between the EU, between member states, how one can do this. I mean, it is within the EU that these clinics are working. So how to do it legally is very difficult, and I think that is something you have to do, also discuss with lawyers. But I think that there is another aspect, and I think that one of my slides at the end was about communication and information to patients. Even if there still will be those that travel, I think that by giving patients and relatives good information about what is possible, what is not possible, why there is no chance at all for improvement with some so-called treatments. I think that many actually then they don't go for this type of treatment when they understand. So we have to find a way of giving good information to people.
Yes, I have also a follow-up question regarding this about safety and efficacy, which I think is very nice with the EASAC report, that it actually says something very practical about this. And my question to you also is, of course, the safety studies will be like a phase 1 clinical trial, but when you're going to plan your efficacy study into phase 2 and phase 3, it is obviously very difficult to estimate the power, how many patients you need, because there's so little data out there. What are your thoughts on this?
Yeah, no, that's a difficult question, and I don't think that I can really say so much about that. When it comes to the that was not discussed at all at this meeting. So it's more. And we are not really, when it comes to the stem cell trial that I'm involved in myself, we are talking about in phase 1, we are talking about 8 patients or something like that. In phase 2, we have not really discussed that, so I cannot say anything about this. I think this is a very important, but I also think that what your question illustrates what I said, that some of this have to be developed because cells are so different from drugs. One aspect which has been discussed in my field alone is whether one should have some surgery control. If you put something into the brain, what would be the best control? Probably the best sham surgery you can do is to just open the skull without opening the dura. But that's of course not a real proper control of what you're doing, because you're actually doing something into the brain tissue. And then if you then want to give immunosuppression, if you have something, an organ or so, you have some want to control, should you give immunosuppression impression also into the sham group with a possible adverse effect. And I think that these questions, and also with power, I think you have to, it has to be, of course, a new type of discussion with regulators and in the respective fields, that we as scientists have a responsibility to, together with regulators, to come up with ideas of how it should be done to promote the field moving forward.
In the report, well, first of all, Olle, in your last slide, you had a fast track.
Yeah.
Pointing towards an airport. And however, by reading the report, I think you are fairly cautious about adapting fast tracks. And like the Japanese, they have for a number of years now developed a fast track system, but still the report is fairly critical, if I understand it and interpret it correctly. So how do you guide regulatory authorities to move forward, since in one hand you want to do this as sleek and fast and with as few hurdles as possible, but still you need to have the hurdles, and you need not to start clinical trials at a too early stage.
No, I think this is a very important issue. I think I have a lot of contacts also with Japanese colleagues, and I think that there is, of course, this problem that shall we take another 20 years before we have something really valuable. I mean, I started to put the first dopamine cells in patient 33 years ago or 35 years ago with adrenomyelite tissue, and we still don't have a treatment. And this is, of course, not this balance. But I think what I said about when it comes to moving the field forward, to find the best solution for this together with regulators. I think that the problem is when companies which have, let's say, they have money for a couple of years, they want to move very quickly to the clinic. And I think that we have to have certain criteria of what is absolutely necessary to show. No one wants it to take longer time than necessary. But there is another aspect on what you brought up, and that is desperate patients. Patients who are, let's say, have just one more year to live, and that there is a stem cell treatment or gene therapy treatment which potentially could help them. But they have not been subjected to a phase 2 or phase 3 clinical trial. So when can unproven treatments be introduced? What will be the requirements of so far unproven treatments to be applied in patients? What are the requirements? And that is something which one can read about in the ISSCR guidelines, and which was discussed a lot in Sweden a couple of years ago. With this, when there is really not more to offer, and that there are some scientific basis to move to the clinic or something. And I think that's an important issue. Actually, it was discussed a lot in Sweden after the Karolinska, what happened at Karolinska. According to Swedish law, you are not allowed to, it should be vetenskap och beprövad erfarenhet. That is what is allowed, nothing else.
You just mentioned that it's more than 30 or 3 decades since you did the first transplantation of cells to Parkinson's patients. And it's also more than 20 years since Jamie Thomas developed the human embryonic stem cells. It's almost 15 years since Shinya Yamanaka developed the induced pluripotent stem cells. And I remember participating at meetings in the beginning of 2000, and everybody was saying that in 5 to 10 years we would have clinically proved studies demonstrating the efficacy in larger trials. We're not there yet.
No.
If you look, based on your knowledge, your history, the overview that you have of the field, how would you recommend Norway to be a part of taking this field forward, knowing that this is a field that takes decades to develop, to evolve and develop?
Yeah, I think that Norwegian science has a strong tradition in some areas, and I know these areas. I don't know about all areas, but I think that the strong science, and to build on the strong science, that is the most important. I don't think one can say anything about how long time it will take. But I think that there has been a paradigm shift during my career. I mean, I started to work in neurology in 1976. And I had my first boss at the clinic, who was the old-style clinical professor. He asked me what I was interested in, and I said I was interested in trying to repair the brain. And he looked at me and said that that was the most stupid idea he had ever heard, because everyone knows that that is impossible. And I think from that point of view, I think that has been my basis for my work. And I think that it's not time to give up. It's now time to put a higher gear, but to base it on solid science. And I think that for Norwegian science, which is very strong, not least in neuroscience, which I know very well, I mean, this is a very good basis. But it will require long-term scientific and clinical effort.
Thank you very much, Professor Olle Lindvall. It has been a thrill to listen to you, and thank you for your contributions.
Thank you.
Then we will make a short break. We will start again at 15:00 precisely. So please be back if you want to get the start of the next session. So a break until 15:00. Vi vil nå få 6 korte innlegg som hver for seg vil forsøke å besvare det store spørsmålet som vi har her. Og det dreier seg om hvordan skal vi gå fremover innenfor regenerativ medisinfeltet i Norge. Åpner ikke for panelspørsmål etter hvert innlegg, så noter de enten i chatten eller på et eget ark, så ikke glemmer de hvis vi kommer tilbake til det. Først ut Rune Kjeken. Rune har doktorgrad i molekylærbiologi fra Universitetet i Oslo. Han har vært postdoktor ved MBL i Heidelberg, direktør hos Innovio Biomedical Company i San Diego, hvor han jobbet med metoder for genlevering og DNA-vaksiner mot kreft og smittsomme sykdommer. Rune Kjeken i dag fagdirektør for avanserte terapier ved Legemiddelverket. Rune, ordet ditt.
Ja, takk for invitasjonen. Og jeg har da fått den litt slitne oppgaven å presentere regelverk, som kanskje ikke det mest spenstige her, særlig etter at dere har hørt disse interessante vitenskapelige diskusjonene, men jeg skal prøve å i hvert fall ikke være for mye detaljert med regelverk. Men dette jo litt komplisert, fordi at i bunn og grunn så har vi ikke noe regulatorisk rammeverk spesielt tilpasset regenerativ medisin, men det vi kan si da, at for regenerativ medisin jo litt sånn dårlig definert, litt vagt definert, det veldig vidt. Men stort sett så kan det meste av det vi snakker om dekkes av det vi kaller avanserte terapier, eller ATMP-er, som består av altså vevsterapi, celleterapi, inkludert stamcelleterapi og genterapi. Og der har vi et regelverk som EU-kommisjonen i sin visdom satte på plass i 2007, nettopp for å få denne type terapier inn under medisinparaplyen. For før så var ikke denne type medisin, eller denne type terapi regulert på samme måten. Det var ingen krav til effekt for eksempel. Det var krav til sikkerhet selvsagt, men ikke til noen effekt. Så ved å regulere det som medisin, så får man på plass da et krav om at det skal reguleres på samme måten, og at det skal også være krav til effekt, som det viktige forskjellen her. Så man skal da diskutere, man må da dokumentere sikkerhet, effekt, for å kunne få godkjent disse typen terapien, og ha det på markedet i Europa. Og man får kvalitetsstandard for produksjon, og man får krav til sikkerhetsoppfølging og så videre. Og kriterier for å klassifisere noe som en ATMP i forhold til, skal si, transplantasjon, det går på to kriterier, enten om det homolog eller ikke-homolog bruk, det substansiell manipulering. Så tar man ut celler i et operasjonsrom av en pasient, reanalyserer i den samme prosedyren, og det homolog bruk, den skal ha det samme funksjon når det blir satt inn som man tok det ut, så sier vi at dette ikke legemidler. Hvis det ikke homolog bruk, eller hvis man har manipulert det, for eksempel dyrket celler, så ser vi på det som legemidler. Da kommer kravene inn til dokumentasjon om effekt. Så hvorfor satte man på plass dette regelverket i 2007? Jo, det var fordi at man hadde store forventninger til hva som skulle skje i dette feltet. Man så at avanserte terapier, det skjedde mye i feltet. Man tenkte at dette kunne gi muligheter for behandling for en rekke tilstander der man ikke hadde noe alternativ i dag. Og så man også i Europa at det var veldig forskjell på ulike land. Det var land som Tyskland, England, Frankrike som lå veldig langt fremme, som hadde et visst tilbud til sin befolkning, og så hadde andre land som ikke hadde det. Så man tenkte at ved å ha et strømlinjet regelverk, så ville man skape et marked hvor man ville gi pasienter lik tilgang. Og så man det som har blitt nevnt her flere ganger, at det var også den gangen en del suspekt bruk av særlig stamceller. Så man hadde allerede den gangen man hadde stamcelleturisme, man hadde udokumentert bruk. Og ved å få plass et regelverk man kunne stille krav til effekt og sikkerhet, så hadde man også et regelverk man kunne gå inn og stoppe den type virksomhet. Så dette var omtrent situasjonen i 2007. Man så for seg en rekke produkter som skulle komme i løpet av få år. Nå har vi sittet i en komiteen for avansert terapi som holder på med dette her i over 10 år, og jeg tror det omtrent sånn vi ser på det i dag. Det vel omtrent her vi i dag. Så i godt driv, men litt kaotisk, og ikke helt der vi trodde vi skulle være for 10-15 år siden. Så hvis jeg ser på produkter som har kommet seg hele veien fram, altså de har gått gjennom kliniske studier, de er på markedet, man kan kjøpe dem, man kan bruke dem som pasient, så det ikke hundrevis som vi kanskje trodde for 10 år siden, det cirka et dusin. Og en annen ting å legge merke til at de færreste celleterapier, eller det vi tenker på som regenerativ medisin, det meste genterapier. Og det som av regenerativ medisin, det kanskje ikke de mest avanserte, fancy produktene, sånn som dere hørte om i dag. Det kan si, litt mer down-to-earth-produkter som for reparere bruske kne, anale fistula og så videre. Og så ser man også at det ganske mange produkter som ikke har klart seg. De har ikke klart seg kommersielt, selv om de har fått godkjennelse. Men det skjer mye fremover, så det grunn til optimisme. En av de tingene vi driver på EU-nivå å gi scientific advice til utviklere. Og det har vært en veldig dette viser at det et veldig stort trykk på feltet, men særlig på genterapi. Det ikke i fiolett så det vevsterapi, og så ser man celleterapi i grønt. Og man ser at det har vært lite utvikling på det feltet når man ser på antall produkter som under utvikling. Og det har tror jeg noe med den enorme suksessen man har hatt på genterapi, for det et alternativ til annen type behandling i en del tilfeller. Og så har man hatt disse skandalene som har skjedd med for eksempel trakea på Karolinska og så videre, som nok har bremset feltet og også folks vilje til å investere i feltet. Og det vi ser, vi holder på med scientific advice til utviklere på EMA-nivå, og det vi ser jo mye av det som ble nevnt i den første framsynningen her, at man har lite moden produksjon av produkter så ofte. Man har ikke helt kontroll på hvordan man lager celler. Det mangler ofte skikkelig klinisk non-clinical proof of concept før man går i mennesket. Ofte så jo dette ting som utvikles for sjeldne sykdommer, og man har kanskje ikke helt skjønt at det kreves en viss pasientpopulasjon. Det ofte litt lite gjennomtenkte studiedesigner. Vi ser ofte, særlig for disse små, fordi det utvikles, tror jeg, av små biotekselskaper og til dels akademika, at man har ofte tenkt lite over den statistiske designen på studiene. Man mangler ofte en ordentlig statistisk analyseplan med hypotesetesting for eksempel. Og man sliter ofte med å finne egentlig informative endepunkter som virkelig dekker den kliniske biten av produktet. Og en annen ting som mange ikke har tenkt på, fordi man kommer gjerne fra et sånt lab environment, at man i en sjelden tilstand hvor man skal gjøre en single-arm eller enkeltarmstudie, så trenger man også data for ekstern kontekst, så man må ofte samle historiske data fra en pasientpopulasjon. Så dette ting vi kommer bort til. Vi driver også og skriver scientific guidelines, vi jo regulatorer, så vi gjør det. Vi liker det, og holder på med det, og det sånn passe nyttig, og vi har skrevet masse guidelines på dette feltet, særlig på celleterapifeltet. Jeg skal ikke gå inn med alle disse her, men det nå likevel sånn at dette et raskt utviklende felt, og det begrenset nytte av disse guidene tross alt. For det nå en gang sånn at dette ikke ting som i stein, det et veldig, veldig flytende område, og sånne guideliner mer en slags utgangspunkt egentlig for en samtale med regulatoriske myndigheter. Så litt om, dette var det som skjer på EU-nivå, og det på en måte ganske ordentlig og greit. Det litt mer problematisk hvordan det reguleres på nasjonalt nivå, for en del ting ligger på nasjonalt nivå, blant annet ansvaret for kliniske utprøvinger, hvor man vurderer kvalitet, non-klinikk og studiedesign og så videre. Og det ofte sånn at de som ønsker å gjøre kliniske studier, de kan shoppe rundt. Så hvis et legemiddelverk i et land litt vriene, så kan man gå til neste land. Og det litt dårlig samkjøring mellom ulike land der, fordi man konkurrerer også til en viss grad om kliniske studier for å ha noe tilbudt pasientene sine. Og så det en spesiell ting med avanserte terapier. Det noe som kalles sykehusuttak, og det noe EU-kommisjonen satte som en slags unntaksbestemmelse når de innførte denne reguleringen, fordi man ser at det en del legemidler som ikke helt passer til et sånn markedsføringsløp. Og da har man et alternativ. Men det altså ikke ment som et alternativ til kliniske studier. Det skal være samme krav til produksjon. Og det skal være ikke-rutinebasis. Så vi ganske strenge på det. Og så har man da selvsagt en slags sikkerhetsventil som kalles godkjenningsfritak, som vi bruker under eksepsjonelle omstendigheter. Særlig der hvor det ikke finnes andre behandlingsalternativer. Så vi gir det noen ganger for eksempel i sistelinjekreftbehandling, hvor man får selvterapi for eksempel. Og så driver vi også med klassifisering på nasjonalt nivå, så vi kan klassifisere ting som for eksempel en avansert terapi, eller ikke. Og det et slags ris bak speil, det noe vi kan bruke aktivt for å stoppe virksomhet som vi ikke liker. Men det samtidig litt vanskelig, det et litt dilemma da, for at en del av disse produktene i grenseland mellom å være medisinske produkter, være medisin, eller å være noe annet som ikke krever den samme dokumentasjonen. Så du har for eksempel behandling som skjer på Radiumhospitalet, hvor man tar ut stamceller fra fettvev for å behandle sårskader. Som vi ikke ønsker å egentlig gå noe nærmere inn på, for vi ønsker ikke å lage vanskeligheter for de som holder på med denne type behandling. Men samtidig så har du veldig lik behandling som skjer i en del stamcelleklinikker, hvor man tar ut for eksempel mesenkymale stamceller fra fra fettvev, og bruker til ting som vi ikke like begeistret for. Men vi kan ha en litt pragmatisk tilnærming der, og vi kan bruke klassifisering som et sånt verktøy. Så bare til slutt, jeg skal prøve å være ganske kort, vi ser på oss selv som, vi prøver både å være en slags gatekeeper, det vel det vi satt til, men vi vil også gjerne være en fasilitator for dette feltet. Så vi har en målsetning om å være med å fremme regenerativ medisin ved å gi regulatorisk bistand. Vi jo et lite land, altså vi har begrenset kompetanse, tror jeg, både på utviklersiden, men også på regulatorisk side. Men vi har, nettopp fordi vi har sett disse produktene komme, så har vi gjennom flere år forsøkt aktivt å bygge kompetanse, og det gjør vi gjennom arbeidet i EMA-systemet. Vi sitter i vitenskapelig komité der, og arbeidsgrupper, og vi deltar i vitenskapelig rådgivning og så videre. Og vi er ansvarlige nå for godkjenning av blant annet flere CAR-T-produkter i hele Europa. Så det måten vi bygger kompetanse på, og vi ønsker jo selvsagt da at pasienter skal få tilgang til nye terapier, men de skal få tilgang først og fremst ved å delta i kliniske studier, eller de skal få tilgang til godkjente markedsførte produkter. Det de veiene vi ser først og fremst. Vi ønsker å være restriktive med å tillate alternative veier til at pasienter skal få bruke regenerativ medisin, som for eksempel sykehusunntak. Det finnes tilfeller hvor vi gjør det, kanskje der for eksempel det har vært en klinisk studie som nå ferdig og har litt erfaring, men vi ønsker ikke å bruke det i stedet for en klinisk studie for eksempel. Og så har vi da godkjenningsfritak som vi gir på case-to-case-basis, og så ønsker vi som sagt å bruke klassifisering som et verktøy til å stoppe uønskede produkter. Det å klassifisere som legemiddel, for da har vi mulighet til å gripe inn. Vi har på en måte ikke vært i den situasjonen i særlig grad enda, og vi litt spent, for vi vet at noen av disse klinikkene som, hvis de etablerer seg i Norge og man stopper dem, så har de en viss juridisk bistand. Jeg tror jeg skal stoppe der.
Jeg skal til å si, jeg må be deg komme inn på landing, Rune.
Ja,
veldig bra. Veldig bra. Da går vi direkte videre til neste innleder, som professor Kamal Mustafa. Mustafa tok sin doktorgrad ved Det Karolinske instituttet i 2001, og har siden 2010 vært leder for forskningsgruppen Styrt vevsbygging og regenerasjon ved Universitetet i Bergen. Professor Mustafa leder nå en av de få kliniske studiene i Europa på avansert celleterapi. Kamal, ordet ditt.
Yes, okay. Thank you very much for the invitation. I'm very happy to share my experience, and as been mentioned, I'm a dentist from the Department of Clinical Dentistry, Faculty of Medicine in Bergen. My research, I'm leading this group, the tissue engineering group, which is relatively one of the large group at the Faculty of Medicine. And of course, natural, since the group is hosted by the Department of Clinical Dentistry, my research is more or less focused on bone and bone regeneration. And my interests actually come from for example, when you see such a patient, for example, comes to our clinic, and we see more than 100 patients annually, actually. They come with very narrow alveolar bone, and where they want to have what we call a dental implant. And we know that the dental implant, the diameter of dental implant is more or less more than 4 to 5 millimeter, which make it impossible to accommodate dental implant in such narrow alveolar bone. And then we saw that and of course, the only possibility for this patient is to transplant the bone from other part of the body. And we know all of us, we know the drawback of the bone the transplant and bone graft. That's why we believe that regenerative medicine, tissue engineering might, there is a demand, and it might help such kind of patients. As I totally agree with the first speaker, and this has been, for me, it's been a long journey. that we have to address all the safety issues and the efficacy when we think about the product. When I talk about product, more or less I focus on autologous stem cells, and we wanted to make new bone for and help our patients. So I started with this project almost like 13 years ago, since 2007, do all the work at preclinical levels in the lab, large animals, and finally we managed to develop a standardized protocol in collaboration with our partner in Germany. And this protocol has been published, and it could be used for expansion of mesenchymal stromal cells. The advantage of this group, or this protocol actually, is standardized. And of course, we could use platelet lysate to replace FBS as a substitute. Another advantage, which is also important, that it's possible within this, by using this protocol, to transport the cells within Europe, between Bergen, for example, to Germany within 24 hours, and it still keeps the viability of the cells and the stemness of the cells. So I think that absolutely with great advantage. So we could share and send the cells between Oslo and Bergen within 24 hours without any problem. Also, we could observe that although stem cells from more or less they have comparable biological characteristics, they are different when we think about their different sources. Different with different functional, they have different functional differences, and also their potential differentiation is different. Collecting all this information, we after some years, we managed to reach the patient and translate all the laboratory's findings to the first patient, where it was possible, for example, to increase the width of this very narrow alveolar bone in a way to accommodate dental implant. So this has been successful by using autologous stem cells derived from the bone marrow in combination with FDA-approved biomaterial. Of course, when we talk about patient, we have to also consider that if the material is approved or not approved. So then this trial was successful. We treated 11 patients, and actually now we have 3 years follow-up. And interestingly, actually, we did not have any bone resorption after treatment. And all patients received a dental implant and their new teeth in the new bone, in the newly formed bone. Based on this, of course, to introduce therapy to the hospital, as we all know, we need to have to expand the trials, include more patients. And fortunately now also we got another funding, and then we are leading also a relatively large clinical trial where 8 centers in Europe in 5 different countries are participating, and we are doing a randomized clinical trial within the Max Bone project. So already being patient is the trial is ongoing, and what we have learned from this trial, because now we've been discussing the regularity issues and how to do that, and we've been in this process also for the last almost 10 years. I do acknowledge the strong support from the Norwegian Medicine Agency, for example, because in this trial, the approval being processed by a process called voluntary harmonization process. And I acknowledge NOMA that they accept to lead this process, because then we had to get the approval from 5 different countries at the same time. This has been challenging procedure, but is recommended, introduced and recommended by the network of European medicine agencies. So we learned a lot from this experience, from this trial, and now the trial is, as I said, is being approved, and patients are recruited, and yeah, it is an ongoing project. where we have 150 patients, 100 with stem cells and 50 with as a control, which is a bone graft, normal bone graft. And in parallel, of course, we saw that we could translate our protocol and target long bone defect, non-healed bone defect in the long bone. And again, also our trial also being approved. And as you could see that one of the examples, this boy is not being able to walk for more than 5 years. And they tried at the hospital to treat the defect with bone graft, and it was several failure. But again, we could see that this is also promising, being successful, and it is also is ongoing trial. So currently we have 4 clinical trials being approved, and all of them are ongoing. In parallel, of course, we are working to develop the biomaterial, because so far we use granules, which might not be appropriate for different applications. But this is only the approved kind of a scaffold. So we're using the 3D printing technology, and we are trying to mimic the compact and the spongiest part of the bone. This is an ongoing project. And again, we have to go through the whole procedure. And currently now we are testing the product into large animals. So hopefully that it could be introduced in a couple of years. Of course, we're combining the 3D printing technology with the bioprinting. And again, this is also a promising approach. It's possible that the cells could survive and the printing process and could survive for up to 3 weeks. That's also been published. This is a lot of work. As I said, I've been working with this almost for 13 years, but large group of people at the Faculty of Medicine as well as the hospital is contributing. I definitely acknowledge all the effort by all of them. My group also is relatively large. All the students, almost 9-10 students and postdocs in total, almost like 18 candidates, are working on one objective: to promote the therapy to the hospital, hopefully in some years. Currently, we are working on 8 projects using mesenchymal stem cell for regeneration. In these 8 projects, 4 of them are clinical-oriented projects, where we have 4 clinical trials ongoing. But again, 4 of them are trying to develop the biomaterial. So I do acknowledge all the funding agencies contributing to our effort. Thank you.
Thank you very much, Kamal, Mustafa. Then we will continue the high pace, and den neste innlederen professor Goran Petrovski. Petrovski netthinnekirurg og leder for Senter for øyeforskning ved OUS. Senteret har bred erfaring med stamcelleforskning og transplantasjonsstudier av hornhinne og netthinne. Goran, ordet ditt.
Thank you for inviting me. I will give a brief overview of the many projects and exciting stem cell therapy possibilities in the eye. I always like to start with this slide to introduce you to probably one of the smallest organs we have in the body, but it's so complex and very important. Basically all these circles that you see here are places where we have isolated and expanded and used stem cells for different cell therapy projects. The report that we got also from Joel is the World Economic Forum says that stem cells could be the medical innovation of this century. And only a few specific indications exist for treating intractable diseases such as hematopoietic and skin tests. I will focus one part on the cornea in this presentation, and the other part is on the back part of the eye, the retina. You can consider these two as a photo camera, the lens and the film part of the photo camera. So regarding the cornea, which is our surface, clear surface of the eye, there is an ongoing transplantation of these limbal epithelial stem cells, which are taken usually from patients with chemical burns from the good eye, and these cells are being expanded ex vivo on amniotic membrane, and this is the eye to be transplanted. And as you can see here, afterwards, when they have deficiency of these stem cells, basically we get to get a clear cornea and restore the vision in these patients. One source is from the good eye of the patient, but we have worked also with other sources with collaborators in Finland, such as iPS-derived limbal stem cells and also embryonic stem cell-derived limbal epithelium. So these are all exciting sources for future therapies. The good side about cornea therapy actually is you're probably thinking that the most frequent transplantation in our body is kidney transplantation. No, it's not. Actually, the cornea transplantation is the most often and most successfully done transplantation in the world. So the cell sources here are corneal limbal epithelial stem cells, which are autologous, and also iPS and embryonic stem cell derived limbus. We also work with cells which can regenerate the stroma part of the cornea. This is the middle part of the surface of the eye, which should be clear, and it contains corneal stromal stem cells. We have actually characterized these stem cells to be mesenchymal-like stem cells with all the characteristics that Kamal has shown with the surface markers, and we are using different scaffolds. This is a collaboration, an example of Scandinavian work with a group in Linköping, where also we're performing also in Oslo a femtosecond laser this clear cut of the cornea, and then we can put these scaffolds which are cellularized into the stroma, and this is actually live imaging of the cornea up to 8 weeks and on. The source of the cells here is can be also autologous corneal stromal mesenchymal stem cells, but there have been attempts with bone marrow MSCs and fat derived stem cells. This is work that actually we're doing in collaboration with Hanne Scholtz group. So another source of these cells is actually spontaneously derived 3D corneal stromas. We have actually generated organoids, which to an eye of a person who knows about histology, it would just mean that they're seeing a similar staining of collagens as we see in a normal cornea here. And these are actually the 3D structures which we get in a Petri dish. In addition, as mentioned, we are using the 3D printer and collaboration with Hanne Scholtz's group. And we're now working on different type of scaffold materials, and then using the same femtolaser technique on pig eyes, providing cellularized bioscaffolds for transplantation into this flap technique that we have. As a retina surgeon, this stays near my heart actually, and this is the transplantation of the retinal pigment epithelium. This is something which has importance for different diseases, which can be also genetic, but here is the largest group, the age-related macular degeneration, which is the leading cause of blindness in the elderly. The aim here is to obtain partial or full visual rescue, or to delay the degeneration, which is done in a pig model and consequently in humans. So in the EASAC FEM working group, there is actually even a mention of these retinal disorders, which they put particular emphasis on this work. And different models of cell transplantation have included cell suspension, scaffold, stem cell derived, such as embryonic stem cells and iPS derived. Our focus in Oslo has been on primary cells and iPSCs, while, for example, the group in Karolinska, they're a bit ahead. They're working with embryonic stem cells derived RPEs, and working on a clinical trial. To give a comparison, actually, they have raised from industry 48 million Swedish crowns. So this is something which is important to get these clinical trials moving. We can't actually do a clinical trial from a Helseservice or Forskningsrådet project. We have an EEA grant on this with Czechia, where we do our pig transplantations. And this is just a glimpse of the pig transplantations. We take this scaffold, which has these RPE cells. We do a procedure called vitrectomy. We put this under the retina, and then we do live observation of the animals up to 6 months to see whether the RPE implant is still remaining under the retina, as you can see it here. And also we do characterization by differentiating the border between the human RPEs and the pig RPEs. So here the source of the cells is primary human iPSCs and ESC RPEs. Another treatment which actually we're doing is using platelet-rich plasma from the patients. This contains actually the natural glue and also the growth factors such as BGF. And this is a patient that has a macular hole. This is the sharp vision part of the patient, and you can see before.
Der falt Goran ut for meg, og det ser ut som at han gjorde det for alle andre også. Da tenker jeg at vi går videre med en gang, og da vil jeg be de som står for det tekniske ta ned presentasjonen til Goran, så vi kan gjøre klart til neste presentasjon. Det var synd, men sånn kan skje. Neste innleder professor Helge Ræder. Ræder leder for stamcelllaboratoriet ved Diabetessenteret ved Universitetet i Bergen. Han har en bistilling som overlege på barneklinikken ved Haukeland universitetssykehus, og også, interessant nok, prodekan for innovasjon. Helge, ordet ditt.
I think I'm going to take this in English since I realize that there are several English listeners here. So my thanks for the invitation. I'll talk about my favorite topic, future potential diabetes cell therapy. And I'll start as a clinician. I'm a pediatric endocrinologist, and I treat diabetes patients. And this is Philip, and he is one of the happy boys playing football with others. But there's one big difference, and that is his insulin pump, and that puts him in a vulnerable position. Without correct dosing of insulin from his pump, he might risk potentially fatal hypoglycemia or hyperglycemia. So finding another solution than the current state of the art with insulin pump is very important, and that's where diabetes stem cell therapy comes in. And there are currently several options which can be divided in therapies with a limited source, current pancreatic transplantations or islet transplantations, and then the promising field of cell therapy with unlimited source coming from autologous, allogenic, and even xenogenic sources. I'll focus on the IPS-based therapy, which has been my main focus, but I'll also show some nice data that we published last year on the future possibilities of gene therapy by repurposing islet cells. So starting with the diabetes cell therapy, there are several challenges, and as was mentioned by Lindvall, the importance of good experimental basis is important also here. And for us, it's the focus to develop functional cells that is really a matter of high importance here. And if you make good functional cells, you will have less aberrant cells with the potential to acquire side effects, including tumor growth. So that is really essential when you want to put these cells into patient. And such studies are ongoing as we speak. BioSite is a company currently putting capsules with cells into patients, and try to highlight some of the potential pitfalls that might occur here. Of course, there are other important aspects, including avoiding immune attack, that I will not cover in this talk. I briefly mention encapsulation strategies with emphasis on allogeneic encapsulation. So in my paradigm, we are using induced pluripotent stem cells, which we acquire by reprogramming skin cells from patients and using chemoscal for the directed differentiation into pancreatic beta-like cells. the basis of our studies is 30 years, 25 years of studies on monogenic diabetes in several forms. In Bergen, we have the Norwegian Moly Registry, and for several years we tried to characterize the genetic causes leading to the findings on new monogenic causes, including cell MODY-8. Or MODY-8 and other forms of monogenic diabetes, which has formed the basis of our subsequent studies. Because using this patient material, we have been able to reprogram the fibroblasts from the same patients into iPS, and using episomal reprogramming, and show that these cells are pluripotent. And by pluripotent cells, we can prove that the cells not only have the markers of pluripotency, but also have the ability to make the 3 germ layers, endoderm, ectoderm, and mesoderm. And the reason why I mention this is that in our attempts to do reprogramming, we tried to carefully study these reprogrammed cells, and we realized that reprogrammed cells are not equal. And this is an important point, because a lot of therapy, not only diabetes cell therapy, but CAR T therapy and several other future cell therapies that are based on iPS, they require that these cells are normally functioning, and we are not seeing that that is the case for all iPS cells that are produced. And a paper that we published last year with Ingrid Bjørlykke, my former PhD student, we show that the typical, in some of the colonies, we don't see the typical colony morphology, and we correlate that to the potential to make the 3 germ layers. We see that the cells we call unstable, they miss some of the typical characteristics of the 3 germ layers. And by proteomic studies, we see that unstable and stable cells, as we call them, when the colonies disintegrate, they have very different proteomic signatures, pointing towards markers indicating EMT, epithelial to mesenchymal transition. That is an important thing to bear in mind. The next step is to direct reprogrammed iPS cells into pancreatic beta-like cells, and that is also not straightforward. We are attempting to mimic a process that takes 9 months in utero, and we have protocols of 1 month. So of course this has a lot of caveats as well, and by a tour de force of several scientists, there are currently protocols that we rely on. Interestingly, in this regard, is the fact that a lot of the informational protocols, it's not only derived from PubMed, but from unpeer-reviewed paper information in Google Patents. That's some of the places where we retrieve that information. That is also a potential challenge in this field, that we don't find published papers. 3 minutes left. Okay, but this is important, and our aim is to make functional cells, and to really prove that we have functional cells is also one of the challenges that we see, because we make pancreatic beta cells with the right markers such as insulin and AKX61, but and with the right ultra micrographic appearances, but they are not functional. They don't produce increased insulin levels when glucose levels goes up. And we also note that some of the cells are not hormonal, only producing insulin, but they are bihormonal cells with insulin and glucagon, which is it's a problem. We try to map the causes doing proteomic studies, and we found that there is an increased Wnt signaling. So we try to pursue that further and try to see if Wnt inhibition could improve functionality, but it did not. It increased number of monohormonal cells, but not increasing the level of functional cells in terms of increased insulin secretion by increased glucose secretion. So we tried allogeneic encapsulation as well, and we also realized, in this regard, we realized that we had an increased yield of beta cell markers, but not increases in functionality. Then we went from in vitro to in vivo and xenotransplanted these cells into immunosuppressed mice, and still not seeing the rescue of diabetes, but again we see increase of key beta cell markers. So that is one of the challenges. Also in this field, it's important when we deal with monogenic diseases to assess the potential for genetic repair. So we attempted to do homologous directed repair by of the mode mutations. And we've been able to see subphenotypes that are rescued. We see at the bottom left panel that mutated cells have aggregation defects, which is corrected in by gene repair, but we don't see an increase in functionality. So finding functional cells is a challenge in the field. This is my last slide, and this is a paper we had in Nature last year for my fellow, for my group members and collaborator in Geneva, showing that there is a potential to repurpose alpha cells. So in this study, we took donor islets and pack-sorted alpha cells, transduced these cells with overexpression of PDX and MAF-A, aggregated the cells, and put the cells back into mice, and we were able to see functional cells. So that points another way to generate insulin therapy. It doesn't need to be bona fide beta cells. It can be redirected or repurposed alpha cells that can be the cells that cause future treatment of diabetes patients. So finally, my time is up, but I would like to acknowledge my research group, in particular Helge and Ingrid Bjørke performed most of the studies, but also many other group members, the funders, and my great collaborators, including the next speaker, Hanne Scholtz in Oslo. Thank you.
Thank you. The next presenter is, as named, Hanne Scholtz. Scholtz holds a PhD in medical pathophysiology from the University of Oslo. He's a senior researcher at the Oslo University Hospital and heads the laboratory for clinical preparation of insulin-producing cells to treat patients with diabetes. Scholtz is also the leader of the research group at the Center for Excellence on Hybrid Technology at the University of Oslo. Scholtz is furthermore a board member of 2 international societies in the field of regenerative medicine. Hanne, the word is yours.
Thank you so much. It seems that I should take it in English. So my topic is cell therapy for diabetes and autoimmune and inflammatory diseases. So just to mention that Oslo University Hospital is the national organ transplant center and the only one in Norway. So we perform liver, kidney, lung, heart, and beta cell replacement either as pancreas or islet transplantation. So that means that we do have the clinical expertise and the patient population to implement new generative medicine approaches. So what we have done, 6 years ago, we was appointed by the CEO of the hospital that we should assembling and generate the regenerative medicine as a focus research area. So what we did then, we tried to identify and clustering all the researcher group facilities and the clinical expertise and technology of Oslo regenerative medicine research. So to take this a little bit further on, we have taken the next step now to implement also cancer therapy and also other part of regenerative medicine expertise in Norway, and established Norwegian infrastructure for advanced cell and gene therapy with the aim to facilitate new international leading research of ATMP and then develop new life-saving ATMP for our patient. So what do we need? Yes, what we have in Oslo, in the Ræder hospital where I work, is infrastructure for developing ATMP products. And why do you need this? Yeah, you need it to ensure product safety. Department of Cell Therapy has been accredited by GMP, and its size is one of the largest clean room in Europe. And you can see from the number of particles what's going on in a laboratory that's a clean room that's very controlled particles, and there is almost no particles inside the laboratory compared to outside, as you can see here by this one. And also there is control of the size of the particles. So what you also need to have in place that we have, you need a place to store and perform the stability of your cell product in order to generate therapeutic biobank and or stem cell biobanks. And this is also a huge infrastructure that we have been developed, and we are able to put into our product. So as I told you that I work with cell therapy for type 1 diabetes, and as you can see in the pancreas, it's only 2% that's the islets of the pancreas. So that means that we don't need the whole pancreas to be able to cure type 1 diabetes. So this idea came from a long time ago, where you can isolate the islets. So when you develop type 1 diabetes, the beta cells are for some reason destroyed by the immune system, and then the patient don't produce insulin, and with no insulin there is no proper glucose regulation. So if you look at regenerative medicine for diabetes, what do we have? Yes, we have the isolation procedure that we currently doing in the clinic today under the transplant program. So we isolate the beta cells or the pancreatic islets from a donor organ, and then we transplant it back to the patient, type 1 diabetes patient, in the liver. What could be do is what Helge Ræder was doing, and his research, it's that to differentiate pluripotent stem cell into beta cells to have unlimited sources. But for both those strategies, there is a need for controlling the immune system. So since you are introducing cells from another human being, and that can be performed either by cell-based therapy and or encapsulation techniques, as shown here by these devices that you can hide the cells from the immune system. So why is this a good model, regenerative medicine, a good model why is diabetes a good model for regenerative medicine? And one thing is that the cells that we need to cure diabetes doesn't need to be transplanted back to its original site to function, which give us a large advantage of where we can place the cells in the body. Also that we do have the very well-defined criteria for follow-up. Like we can measure the insulin requirements for the patient, HbA1c, glucose control, and the function by secretion of human C-peptide. So we can really follow the product in the human being. And also that we do have for almost 20-30 years now a proof of principle for a cure for type 1 diabetes by islet transplantation. And just 2 years ago, this phase 3 clinical trial that we were involved in showed that it's safe and effective and successful to replace cells in patients with type 1 diabetes by islet transplantation, which is classified as mini organs. So the other program that we are developing in our research, it's the adult somatic stem cell or stroma cells for regenerative medicine. And this is based on, Kamal was also talking about it, and also Helge Ræder and Goran. And this is undifferentiated cells found in tissue and organs around, and their normally function is to maintain and repair the tissue in which they are found. But we can take advantage of these cells because it's possible to harvest them from starting material like the bone marrow, adipose tissue, placenta, and then you can isolate and purify the cells and do a cell expansion to mass produce them into a therapeutic doses. The cells need to be quality tested and released and frozen as a cell product. And we have done this now for the placenta-derived decidual stroma cells. And we are currently applied for acute graft-versus-host COVID-19 RDS, or lung insufficient complication of COVID-19 infection, and also for type 1 diabetes to go into clinical phase 1-2 trials. So the 3D printing is also something that we have implemented in our research. And the reason is because we have small number of cells, only 2% of the pancreas that's containing the pancreatic islets. And we want to place them in the body in a safe way. And one way it could be to generate a scaffold of the pancreatic cells together with some supporting repair cells as the adipose-derived stem cells. And we can do this in a biomaterial scaffold that also Atala was showing you a great inspiration talk about. So in this, we are on the preclinical stage, but this is something that we foresee that can be a reality for into the clinic by having these primary cells together with a biomaterial and make a scaffold by 3D printing technology. So lastly, I just want to briefly skip into another area that's, it's not exactly regenerative medicine translation into clinical trials, but it's something that will support regenerative medicine in the future, in my opinion. And this is the organ-on-a-chip research, which the traditional is that you have in vitro system or you have animal system to be able to go into human clinical trials. What we think that one of the advantages of organ-on-a-chip platform, where you mimic the human physiology in a chip platform, in one way to get a more physiological readout of the real organ, could be one replacement that could make our preclinical research model for regenerative medicine more efficient than previously, where we needed to go through animal models. And we have a Center of Excellence at the University in Oslo, led by Stefan Kraus. And in this, I think that we can learn a lot of both the developing of organoids and also to look for interaction of the drugs for potential before going into human clinical trials. That's also Atala was saying. So lastly, I just want to briefly say that that I think it's essential, and that's why we are here and discussing today. It's the challenges for bringing ATMPs to clinical reality, and there is issue and challenge that we need to solve before we can put the cells in a clinical reality. And we want efficiency and generate enough cells. We need the cells to be safety. We need to find out what kind of cells do we deal, what is their mechanism of action. These cells need to show genomic stability. We don't want to generate tumor in the patient. So the principle is that we don't want to generate new problems in the patient by curing patient with cells for another problem. And of course, the regulatory submission preparation is huge and something that we need to work together in order to be able to continue this process. So with that, I just say thank you.
Thank you very much. Then the last but not least, Professor Joel Glover. Glover holds a PhD in neuroscience from the University of California, Berkeley, and is a professor at the Faculty of Medicine at the University of Oslo. Glover has been the head of the National Center for Stem Cell Research since 2008. Joel, the word is yours.
Okay, so from experience, one always knows that time runs out towards the end of a session like this. So I'm going to have a relatively short presentation. I'm not going to present work from my own laboratory, which is focused on the same kind of organ-on-a-chip technology that Hanne just mentioned. It's not really regenerative medicine in a clinically oriented way, but rather a preclinical approach. And in our work, we make in vitro models of neurological diseases. So we make basically spinal cords and brains on a chip to study genetic diseases of the nervous system. But rather, I'm going to present something that I think is a more general contribution to the overall effort in Norway, and that's an initiative that we've recently taken to establish a biobank of human iPS cells derived from a few individuals, but that can be used for many individuals. That is a bank of allogeneic iPS cells. And just to provide a little bit of background, since iPS cells have been mentioned a number of times here, but we really haven't gone through the process that carefully, I just want to remind everyone that iPS cells, induced pluripotent stem cells, are derived from fully differentiated cells that you can harvest from the body. You take a biopsy from an individual. In many cases, it's a skin biopsy because that's relatively easy. Then you have a donor somatic cell, fully differentiated cell. That cell is taken through a process called reprogramming, in which a number of genes that are associated with pluripotency are reactivated, so that the cell, the differentiated cell, is in essence spooled back to the stage of pluripotency. You then wind up with cells that are able to differentiate further to any cell type in the body, just like embryonic stem cells can do. And these are called induced pluripotent stem cells, iPS cells. The iPS cells, as I said, can be differentiated in vitro or in vivo to, in principle, any cell type. And they can also be subjected to manipulations such as gene editing. So if you were to take a cell from a person who had a genetic disease, you could, at the stage between iPS cell and differentiated cell here, carry out gene editing to remove the genetic defect, if desired. Now, once you create these different cell types from the iPS cells, they can be used for various purposes: in vitro disease modeling, drug screening, animal transplantation studies, and so on. But they can also be used to treat patients. And there are two different pathways to consider here. One is that one has taken a cell from a patient, created iPS cells from that cell, generated cells that that patient needs, and then you transplant them back into that patient. And this is called autologous cell therapy, which is what Tony Atala was speaking about earlier. The alternative is to take those cells derived from this individual and use them to treat other individuals, allogeneic cell therapy. So that's the main focus of this biobank that we're starting to make, a bank of iPS cells from a few individuals that can be used for other individuals. And the basis for this is comes from an initiative that came out of Japan with Shinya Yamanaka, in whose lab iPS cells were first generated, and who has been spearheading the use of iPS cells both as research tools and as clinical treatment options. And the point is that we have a system of cell surface markers called the HLA systems. It's steered by a set of genes that determine the nature of these surface proteins. And it's a very elaborate system that basically gives all the cells in the body of one individual a certain address, molecular address, so that the immune system of that individual can recognize its own cells and cells or cells that might come from another individual that would not have the same markers. So this is called an HLA genotype or a phenotype that each of us has, and which encodes all of our cells so that they can be recognized by our immune system as belonging to us. The problem arises, of course, in all examples of transplantation. What happens if you put a cell from one individual into another individual? Well, the immune system in the other individual will recognize those cells as foreign and will start to attack them. However, you can find individuals whose genetic makeup in the HLA system is such that they have fewer combinations of these different markers. And for that reason, their cells are more easily transplanted into other individuals without triggering an immune response. So in the example that's schematically presented here, we have an individual who is homozygous for 3 different HLA haplotypes. And because of that homozygosity, those cells will be able to be transplanted into this individual who shares one allele of each of those haplotypes, and this individual, which also shares one allele of each of those haplotypes, but not this individual, where the matching is not as good. So the point is that cells from individuals who are homozygous for multiple HLA alleles have increased compatibility for transplantation to multiple recipients. And it turns out that there are certain alleles in the HLA complex that are most important for this matching. class A, B, and 2R are the major haplotypes that are important. So if you find individuals that are homozygous for these haplotypes, those individuals will be able to share their cells with many others without triggering a very strong immune response. So why do we want to make, oh, and I should say that those individuals who are homozygous in this way and who also have blood type O, so that they match in terms of the ABO blood type, are called super donors for this reason. So why do we want to make a bank of super donor iPS cells in Norway? Well, this is based on the fact that if you try to treat each individual with iPS cells from themselves, that takes a lot of time. To make GMP compliant iPS cells, the reprogramming procedure involved takes many months. So by having a bank of cells that can be used on many individuals, you save time to treatment. You also save cost because fewer iPS cell lines need to be generated. You end up with a biobank of readily available iPS cells that are genetically appropriate because you're taking these from a population in Norway that is relatively homogeneous. So it's easy to find a few individuals who can cover a large segment of the population. And in this particular biobank, we have found 9 super donors already who have the genetic makeup in their HLA system that allows them to cover the needs of almost 70% of the Norwegian population. And then you also have an opportunity to cooperate with other countries who are interested in making their own iPS cell, super donor iPS cell biobanks, and then you can cooperate in order to cover even larger segments of the population. So Ole Johannes told me I have less than a minute left, and this is the last slide. This has led us to initiate a program in which we are starting to reprogram iPS cells from these 9 super donors that we've identified. And this is an initiative that's been spearheaded by the Norwegian Center for Stem Cell Research in collaboration with the Norwegian Bone Marrow Donor Registry and the Ex Vivo Clinical Cell Production Facility, one of the GPM facilities at Oslo University Hospital. And we'll be sharing this bank once it is established with Haukeland University Hospital in Bergen, which also has a GMP clinical cell production facility, so that these cells can be used for future clinical trials and for future preclinical research. So basically, this is a sort of anticipatory project. We're anticipating that there will be a need for this sort of resource in the future, and we'd like to make that available as soon as possible. Thank you very much.
Thank you, Joel. Then we are entering phase 3 of this seminar, the panel discussion. And as you all know, and Hanne even alluded to it, it's the main question is what should be Norway's strategy for regenerative medicine? I have sent out a few questions to the panelists ahead, but bear in mind that all of you that are attendees, please raise your arm or sign in the chat, and I will I would love to hear your views on the questions and the comments on the presentations as well. So, dear panelists, at the start of this seminar, we heard 2 excellent presentations depicting both of them from their various point of departure, where the field is today. We also saw the picture that Rune Kjeken used of the horses that did not make it over the hurdle, or they were crashing and smashing into each other. And of course, the field of regenerative medicine has been partially a field of hype, but also at least not a field of the success that we would love and also anticipated. I want to hear from each one of you shortly or briefly. How do you look at the field? How fast is it developing? And not in Norway, but on a European and North American scale, but also to mention Asia. So how is the development within the field of regenerative medicine? I would like to first give the word to Professor Morten Moe, whom we have not met earlier in this webinar. Morten has a PhD from the University of Oslo. He's head of the clinic for head, neck, and reconstructive surgery at the Oslo University Hospital. as well as a professor of the IDUL department. Morten, you go first.
Thank you very much. I think it was a very fascinating 2 lectures and also a very constructive report, I must say. And I think the field is now moving into another kind of phase. It's not only about safety studies, phase 1 studies, but people are really now starting to design efficacy studies, phase 2 and studies. And it's also focusing on the patient perspective to include the patient-reported outcome measures. So I think this is the way it should develop. And we have also heard how important it is to both publish negative results and positive results. And this way we can move into the clinic, because this is what's really now needed, is to have the focus on efficacy, not only safety. Safety, of course, this is the main pillar, but this is the obvious continuity. So that's one thing I really appreciated.
Hanne, and then Helge.
Yeah, I totally agree with Morten. One thing was what I was thought of when Atala was showing this very nice interaction with the FDA the whole way from the start, because they pioneered a lot of new treatment that we kind of maybe back a little bit up. Wow, have they already transplanted this bladder, or had already done that? And I think closer interaction with the regulatory authorities in Norway, and also with EMA, of course, and FDA, I think from the Norwegian perspective will be important to do, because then we can both go further on to efficacy studies, that also Morten was said that this will be the next step, but also to find the path going forward.
Thank you. Helge, and then Goran.
I think working in a more pre-GMP part of this field, I feel the main constraint is to secure funding for the basic research for stem cells. The reason why I say it is that the more I go into stem cell research, I see there's a lot of caveats, including the variability of iPS cells that I showed, the problems with getting functional cells. These are issues that Lindvall mentioned, the stem cell variability, which affects the safety. And I think it's important to have people in the field that can know all these caveats and can go to the conferences of ISSR, and because these conferences, they develop guidelines and keep the kind of scientific level high, and securing that we have people in Norway that are well aware of these standards, I think is a very important point. Another important issue, I think, is the transparency of science, because what worries me is the publication, or the non-publication of results, especially in the interest of the commercial side of this. And what I see in the very competitive diabetes field is that the withholding of information and the retrieval of information using Google patents, which includes non-peer-reviewed information, that is, in my eyes, a threat to the future.
Goran, and then Kamal. Goran?
Thanks. I was cut out, so not by intention. It was fantastic discussion. I think we need to pull up the sleeves here in Norway. And I'm lacking actually sort of a GMP facility or a way how we will have help with the GMP regulative, so we can move forward some of the trials that may have to happen at the hospital or national level. And also animal facilities where things can be done in a standardized way. Back to Joel's actually super donor issue. That's very important to build in the country, and to continue the stem cell center, because that's something that we can all refer to as a reference point. So, and the last thing is more collaboration with industry. From example with Karolinska, actually, how much money they have raised for retina stem cell transplantation is just amazing. We can't work with small bits of 3 and 6 million crown projects to build a clinical trial.
Kamal and then Joel.
Very nice presentation. I think my main concern, because not a concern, because I have been in experience communication with regulatory bodies, which is very important. I think that we need to communicate more with regulatory bodies in Norway as well as within the European level, because I've been conducting or running this VHP, and that was really challenging process, because at the end it's been approved by the Norwegian authority and been approved by all participating countries. But in the middle of the projects, when I've been discussing with the EU Commission, there was one reviewer from another country which not participating. They come with another view. I mean, some other guidelines, and that is always, even if you are trying to harmonize the guidelines within Europe, still there are discrepancies and different. So that has to be really discussed. I mean, at different level. For Norway, I think I think we should absolutely strengthen the collaboration between the different groups in both basic level as well as the clinical. Basic, of course, I could see that Hanne are working with bioprinting. We are doing bioprinting and we are doing 3D printing. And we know that with 3D printing, bioprinting technologies, there is a lot of different technology, different parameters, and we need to harmonize. We need to at least to complement and work with each other, because it's very difficult to have everything in one laboratory. So I think that should be the way that we should take it further.
Joel and then Rune.
Yeah, I think everyone has made a lot of important points that I totally agree with. I think one of the main issues, of course, is about regulation, and we have to recognize that there are other countries that have sort of gone ahead and have progressed further than we have on this issue, particularly the US and the UK, and even Sweden. Of course, we've heard from Olle that they've been dealing with transplantation of cells into patients for over 30 years now. So they've done a lot of work and discussion focused on that issue. And I think what we really need to do is draw on that experience. I think the EASAC report is something that has prompted us to think about this more closely. So instead of just working within Norway with the Norwegian Medicines Agency, I think we should start to draw on the expertise of other countries. Other than that, I think we have to remember that we're in for a long haul here, and the granting agencies and the politicians don't have the kind of long-term horizon that is appropriate. So we have to think of ways to work within that system, and collaboration between regions and institutions, I think, is going to be key. You know, if you look at cancer, they get a lot of money, but they've been getting a lot of money for a long time. And even when I was an undergraduate, which is getting close to 40 years ago, well, more than 40 years ago, cancer was getting a lot of money and doing a lot of effort towards curing cancer. And they're still not to the point where they're curing all cancers. So, but nevertheless, the money has been coming all of that time. So we need to convince people that this is equally important, has just as much potential for medicine in the future as that effort had when it started way back in the 1960s.
Rune?
Yeah, I think most have been said, and I guess I come from a different angle. I don't see the basic research anymore. I see products when they're, on the borderline of doing the first clinical trial, maybe. So that's where I observe the field. I just remember when I came from biotech myself, I worked with gene therapy, and when I came to the regulatory side of it, I was shocked how sort of it was like, the 10 years behind. And only now the last few years, I think we've seen really what we saw 10 years ago in preclinical research really materialize into useful products for for patients. And I guess maybe to some extent that is where this field is now. So it's like Joel said, you're in for the long haul. But I guess Norway is a very small country. I mean, there's some strength in numbers. So I guess that is something that is challenging, because I think you need to have certain good environments, and especially because if you want to transform this into the clinic, you need to have some, for example, places to manufacture these products. So to build these facilities and strengthen these environments, I guess that's really key. And also I think, well, there's been discussion on not moving too fast, looking after safety, of course. But I think there's a fine line there, because if you wait too long to go into clinic, I think you stretch when you go from animal to human research, because you need to have things in place. So for example, for manufacturing, I think to transform into the clinic, I think that does a lot for the people involved in these projects, actually.
I think I would like to stop you there, Rune. Thank you. Let's focus on the ambitions. What kind of ambitions should we, or you guys, as the principal investigators within regenerative medicine in Norway, what should be your ambitions, or at least the ambitions you would like that Norway should embrace? And when you answer that, I think you should think about what kind of infrastructure do we need, what kind of collaboration within Nordics, within the Europe, within the US, and so far do we need? What kind of private-public partnerships do we need? What should we do different, or what should we accelerate to reach a much higher level in 10 years from now, for instance? I'd like to start with you, Hanne, and then go to Helge.
Okay. I think that it's really important to have an infrastructure that covers all the way from pre-GMP to GMP, because only then you are able to cope up with all the regulatory requirements. So this is what we have done now in this infrastructure application for the Research Council, where we have applied for advanced cell therapy and gene therapy infrastructure. And this should not be covering basics research, but it should covering all the other things. So you are able to get into clinical trials or to the market with your product. I think it's extremely important in Norway, I see that we haven't been so good at, but it's better, but have this industry academia collaboration, because then you are able to get to the next phase with your product, scale up, scale out, because that may be not impossible in the academia, where there is so much pressure of so many different products. And this is also, I think, like Joel say, how should we go out to the public? How should we go out and get the regenerative medicine field be as hot as cancer? And this is something we need to think of, because there is so many diverse disciplines and diseases and conditions. So to be able to merge those, so people said, yes, regenerative medicine is so important to put your money in, then I think we have a job to do there.
Helge and then Goran.
Okay, so ambition starting from the kind of individual researcher's perspective, I think it's important to carve out your own niche, and we should be innovative about the ways we deploy this stem cell research. And I was a little inspired by a talk on the life science meeting in February here in Oslo, when the AI company Sensign in Oxford talked about how to merge big data from NHS with AI and use iPS cells as an in vitro system to prove effects. That is a new way to think about the use of iPS cells. And we try to deploy that principle in Center of Excellence applications now to create a Center of Endocrine Systems Medicine Center here in Bergen, with the aim to merge big data and using AI and IPS models to verify effects. So I think finding some niche systems where you can get stronger than the others that will attract people from the rest of the world. So that's more on the person, the individual level. On the societal level, I think it's important to keep focus. The Norwegian Research Council funded this stem cell program, and then this program was ended and was swallowed by behandling treatment, but with the promise that the program should continue. But I haven't had the impression that they followed up on that promise and really keep doing the lobby work to get the Research Council focus on the promise of regenerative medicine. It needs to be done, and we are the people in the field, so I guess we are the people who has to do it.
Goran and then Joel.
Yeah, I think on the Tony Atala gave us an idea when he talked about the integrated place where all the scientists and the clinicians are meeting. So you talked about what's the infrastructure we need. So maybe the new, at least in Oslo, the new life sciences building where there's going to be, you know, the health part and also the basic science part, that may help out to bring all these groups together. So there will be more rapid development of clinical trials. The other side, which is sort of missing here, is Norway is big on voluntarism, but in the US they're big on philanthropy. And I think there is a lot to do here also, which philanthropy is in a way, gifting for medical science. And they can raise fantastic $80 million in the eye department in Denver to do science. Now that can move mountains.
Joel and then Rune.
Yes, I was going to say on the just to follow up on what Goran ended with there. You know, the amounts of money that could make a big difference here are relatively small in a national perspective. I mean, to make this allogeneic super donor iPS cell bank is going to cost on the order of 40 million kroner, which I think is about the same as 2 parking spots in the parliamentary parking garage. You know, so if we just manage to get some people to look at this with the right perspective, I think people would agree that we would be able to get some money that could help out a lot here. But I think one of the two main problems I see are too little international collaboration. I mean, all of us are involved in international collaborations, but I think especially when we get to clinical level things, then we really have to focus on that. So we have to have this harmonization of regulatory requirements. You know, there's a lot of work that has to be done at a multinational level before we can get to this. I think Kamal presented a great example where he's got a multinational clinical trial going. This is the sort of thing that I think is going to be important because we are a small country, and with this population, it's going to be very difficult to mount clinical trials that have any, power and that where we can trust the results. And the other part, I think, is also an intranational issue because Norway it's difficult to generate initiatives that bring different regions together in a strong way. It's always been difficult, and I think we need to do something about that. I think this field is a place where we could really do that. There are, of course, we have lots of interactions between Oslo and Bergen and with other environments around, but I think we need to strengthen that effort. If we combine forces in an optimal way, then I think we're going to be much more powerful.
Rune and then Morten.
Yeah, I don't know if I have so much to contribute in this discussion. I mean, I'm on the regulatory side. I don't have that much ambition. I guess from my side, the ambition is that we on the regulatory side, we shouldn't be the weakest link. And so we need to keep up with the field and so that we can be useful for you. If there's anything I would like to say, just latching on to what Joel said, I think that's there's a problem that you're going to go and talk with the regulatory agencies in different countries, and you're going to get different advice, and especially because you're going to do clinical trials in rare diseases, you need to do multinational trials. So, and I think that the practical solution for that is actually go to EMA Central and talk to them instead of shopping around. And it's just a better way to do things, I think.
Morten?
Yeah, I think it's all down to find out where we can succeed and create excellence. I think we can use our unique patient registers to identify, do we have the patients? And then we have to identify where we can team up, basic scientists, clinicians, biomedical experts, artificial intelligence expert, industry, and also regulatories and clinical trials units to identify these 8-9 fields where we can really make a difference and heal patients with serious medical conditions. Yeah. So I think we have to find patients, make the best team, and then get funding for perhaps some limited number of projects where we're really going in to solve the clinical problem.
A question to you, Morten, and probably Goran as well. I mean, as we speak, we have the C-19 pandemic. That means that in almost throughout the world, at least the developed world, there is almost impossible to start or to continue clinical trials. However, in Norway, we are running at almost normal speed. Have you been in contact with the industry working on eye diseases within the regenerative medicine field to try them to relocate their clinical studies to your department?
Yeah, well, we have also had to modify a little bit. So we are continuing clinical trials that are ongoing, but we have also, halted the clinical trials that have not been initiated. But we know it's a big problem for pharmaceutical companies. But we have to put patients' security first. So we are also holding back a little bit.
Okay. Goran, do you have anything to add, or was that sufficient, I guess?
No. Yeah, thanks.
Rune?
Yeah, just to comment on that, I think the situation we are in Norway when it comes to clinical trials is basically two things. We don't have many clinical trials in Norway, compared to other countries. And we haven't been struck that hard by COVID-19. So I think that explains most of these differences. Thank you.
Good. I would like to, we have now talked about that it's a fast-moving field. You need to be in it for the long haul, as Joel depicted. We haven't talked about the infrastructure and what we need to do. It has been mentioned: GMP, international collaboration, the AI, patient registries, and maybe to focus the effort. Of course, in a situation like this, it's usually been pointed towards lack of money. Being from the money side, I think I would encourage you to think not about money as a limitation, but instead of comparing parking lots and money, try to describe what you would like to achieve, and try to make up that house of cards, and then fill it with information and knowledge, and make it more solid. Then my question to you guys is, given that level of ambition, what should you do as an individual researcher or clinician, and what should we do together? How should we collaborate in a better way to succeed even more than what we have done so far? I'd like to start with Helge, and then Goran afterwards. You have to unmute, Helge.
Well, as I said, I think finding new innovative solutions, build a new house of cards. We've got to build a new structure, new ideas, new combinations using artificial intelligence and big data new models. That could be one way to go, at least on an individual level. The way on the societal level, I think it is important to have representation, as I said, on Norwegian Research Council programs, but not only there, but in the EU processes, participate in the panels in ERC, and try to influence on the importance of your own research topics. I think that is something that could be important.
And then to you, what should you do?
I think we've been quite lucky this year, actually, with the COVID on our side, with all these EEA grants and possibilities, although there are smaller bits and pieces there. What the Forskningsrådet maybe can do is also see a pattern of certain developments. So we have, for example, on our side, cornea stem cell project with India, then we have with Czechia, retina pigment epithelium. And what I'm more used to, I also worked in part in the US, is that when they see such a pattern, then you get supplemented. When you see a trend, you give more push for that trend. And I just take an example, for example, the Big Med project. I've just read about, the 150 million being pumped into such a project. That's a great activity. I'm just thinking that had we had maybe 40 or 50, we could have probably reached one of these projects to a clinical trial. So it's important to focus on certain areas.
Joel and then Morten.
Not that easy to answer that question, actually. I mean, I think public awareness and lobbying is going to be crucial here, and that's a big job. I've tried to do that as much as I can, and obviously more of that needs to be done. And I think also increasing efforts to coordinate throughout Norway this kind of activity. I think those are the two things I can see myself contributing to moving forward. We really have to get those two things moving well in order to get anything else to function well.
Morten and then Hanne, and be aware of the question. What can you do as an individual together and the society? So Morten and then Hanne.
I think from my perspective and from the hospital perspective, I think it's important to build a clinical infrastructure that can implement these collaborative regenerative medicine studies. And I also think patient information is very important for us as doctors. We have to give them the right information about what works, but we also need to give them information about what things in regenerative medicine that does not work. I think also that is our responsibility.
Yes, I totally agree. And from my point of view, I think we need to do things that we normally don't get credit for. It's not like we can be paid by our research. We need to accept that we need to do something that can benefit more people and not only our own research topic. And if everybody's doing a little bit, then together I think we will be a great team. Also, you need personnel to, what I do is I lead a conversion environment from Oslo Life Science, which is tremendous work to do because it's different faculties from the university. And I didn't know it was so much work, but this is also bringing new innovation and ideas that Helge is talking about. So you need to, so in my point of view, it's need to try other things that you haven't tried before and give a little bit more of yourself.
Then we are getting close to the end of this seminar. We are set to end 4:45. So I would now like to invite each and one of you to have some closing remarks, your take home. You can say more or less whatever you would like to do. So I would like to start with you, Goran, and then go to Joel afterwards. Goran. You have to unmute. you are.
When it comes to our side and the ophthalmology, I think there is a lot that has been done worldwide with embryonic stem cells in IPS and also with even primary cells. So we believe that we have a lot of the infrastructure here. We just need a small kick to get some of these into clinical trials. And the cornea and the retina on our side look extremely promising. So that can put us on the world map in this research. So this is the area I would put the focus into.
Excellent. Then we're gonna save Joel for last, and then go to Rune, and then Morten. Rune, some closing remarks.
Yeah. Thank you for this seminar. It's been very interesting to get a little bit more insight in what is going on. And I just understand by being here that there isn't that much interaction nationally between the environments that develop these therapies and us as the regulators, at least because many of the products I heard today is new to me. So I would just invite everybody to have closer interaction with us on the regulatory side.
Thank you, Morten, and then Hanne.
I just want to say that I was quite inspired by this EASAC reports. I think it's a good document to put us for the next levels needed in this field. So I just want to thank those who wrote that report and also this very inspiring meeting. Thank you all.
Hanne, and then Helge.
Yes, I think this collaboration is a key point here, that we open up and being transparent. I think this will be something to take home, and such a report that we have been discussing today will help us to do that. So in my research field, I will continue working with diabetes. Already do a lot of collaboration with Helge and also with other Goran and Morten with 3D printing. So I think continue with that one, and also continue the Nordic collaboration that we do in the transplant field. We have really Scandinavian transplant groups that share organs and SOPs and infrastructure. So I think learning from other fields will be important.
Helge, and then Joel. Helge. You have to unmute.
So I try to end in an optimistic tone, and I would say that what gives me some hope is the effects of coordination that also Joel and several others have alluded to. So six years ago, when a couple of scientists, including myself and Kamal and some others, we made that initiative towards the GMP facility in Bergen, and just by coordination, we were able to push that effort that ended up in a facility. And now in the NFR infrastructure program, I suddenly got the invitation from 3 initiatives in Oslo, including Joel here and Hanne with Stefan and Kalle Malmberg. And we saw that the concerted efforts of others, that was not only a kind of way to position ourselves to get funding, but also a nice way to meet our fellow researchers. And we had also a conference in Bergen, annual retreat meeting, and meeting sort of people from Oslo there. And I think concerted action is important on a national level, and we are aiming getting there. But we have to also, of course, do it on the international scale as well. But I'm optimistic.
Then I'll leave the last words to Joel. Joel, please.
Yeah, so it's great to end on a note of optimism, and I think given what Rune said, I think we see clearly some things that we can improve here. So that means that this session was definitely valuable for us sitting in the panel. I hope it was also enjoyable and interesting and valuable for those who listened in. The audience is now down to about the same size as the panel. So I'd like to thank everyone who participated, the speakers who are still with us and the speakers who have had to leave for their efforts, making it a really nice seminar, in my opinion. And thanks to you, Ole, for moderating the entire thing, and thanks to the audience for listening in. I think it's been a very positive experience all in all, and clearly gives us a lot of things that we can discuss further. So with that, I think we can call it an evening. And of course, I'd like to thank the Norwegian Academy of Science and Letters for sponsoring the seminar. Without them, this wouldn't have been possible. Thanks again to everyone, and have a nice evening.